One labelled gel permeation chromatography, that's for today's lecture, and the other which says questions for workshop. And the workshop will be next Monday, in here at this time. And the idea is to have a go at these questions before the workshop. In the workshop we'll work through the answers. That'll only be useful if you've had a go at the questions. It's very easy to sit there and look at me doing something or see something on the screen and think, yeah, I get that. But it's only when you try something for yourself where you really work out whether you got it or not. So these questions, have a go at them before the workshop. Then you can see if you got it right or if there are things you need to ask questions about. You can already answer the first two questions. At the end of today's lecture, you'll be able to answer question 3. And if you turn over, there's a question 4. You'll be able to answer that at the end of tomorrow's lecture. So you have until next Monday, have a go at that. Bring your answers to the workshop next Monday. We'll go through the answers. And if you've got specific questions about that or anything else on the course, that's your opportunity. And then next Tuesday, someone else will be here to move on to talking about polymer synthesis. So 2 bits of paper, handout for today, and questions you can have a go at before next Monday. So if you remember last week we were talking about the question of how we account for the fact that, or describe the fact that polymers are generally mixtures. So when we have a sample of a polymer, we have samples of molecules of different sizes, all mixed together. So we have to think about molar mass distributions, and we saw there were different ways of describing those distributions, in terms of numbers or in terms of masses. And then, of course, if we have a mixture and we measure one value, what we have is an average, and we saw that there were different types of average, a number average and a weight average in particular. But then, last Tuesday we were rudely interrupted by the fire alarm, where we were starting to talk about the different experimental techniques we can use to get information about these things, molar mass distributions and average molar masses. And although we didn't go into much detail because we gave up because of the fire alarm, I did mention a technique which is used to get information about the whole molar mass distribution. And that's the most useful thing. That's what we'd really like to know about. And so that's what we're going to talk about today. So we're talking about a technique which can be called gel permeation or size exclusion chromatography. And here's some equipment that sits up on the first floor that's used by MSc students and actually even those of you doing the third year undergraduate project might well put some samples in for this equipment. And like a lot of equipment today, it's quite boring because it's a box or a number of boxes with a computer attached. And what's really interesting is what's going on inside those boxes. But inside one of the boxes, this little box here, there's a column. And that's the heart of what's going on, as we'll see in a moment. Okay, so you should all have met some sort of chromatography before, is that right? Have you all heard of chromatography? If nothing else, one of the first things you do when you're really young and you start to learn about science, you've probably taken a dye and put it on a piece of filter paper and saw or a bit of ink and seen how the colours separate out if you put a solvent through it. Have you done that? Some of you have. Chromatography, that's what gives it its name, chroma, colour, graphene to write. And so typically you have some sort of a mixture, you dissolve it in something which we call a mobile phase. It could be a liquid, it could be a gas. It's passed through or over a stationary phase, which can be in all kinds of forms. It might be a thin layer of something, it might be little beads of something. And in most forms of chromatography, you separate things on the basis of the different affinities for the different phases. So if something really likes the phase that the stationary phase, it doesn't move very fast, and it's separated out from things that like that phase less. The technique we're talking about today is different. The apparatus may be similar, there may be things in common, but we're not basing our separation on affinity with the stationary phase. And so we need to understand what the difference is, why there's a difference. But this is something that many of you will have done in the laboratories. It's thin layer chromatography of chlorophyll. And in this case, it's simple. This is affinity chromatography. We separate out on the basis of different affinities for the silica or whatever that thin layer phase is. But what we're going to talk about today is a kind of column chromatography. It means at the heart of it, we have a column packed with something. It's a form of liquid chromatography. Our mobile phases are liquid, so in that sense it's like HPLC, high performance or high pressure liquid chromatography. But here we're not separating on the basis of how much our molecules are like a stationary phase. We want to separate purely on the basis of size. And here's the thing that most people don't get at first, because with most sorts of liquid chromatography, if you put a polymer into the column, it doesn't go anywhere at all. Little molecules move quite fast. Big molecules, because they generally interact strongly with any surface, don't tend to go anywhere at all. But in this technique, it's the big molecules that move through the column fastest. So the big molecules, the biggest molecules come off the column first, and the little molecules come later. And we have to ask ourselves, why is that? What's going on inside this column, which means that the big molecules apparently move much faster through the system than the little molecules. And what's important to us is that if we calibrate it appropriately, we can get information about the molar mass distribution of a polymer sample. And before this technique was invented, it was a really difficult thing to work out the molar mass distribution. Everyone knew it was important, but although there were experimental methods, and we briefly through the fire alarm mentioned them some of them last week, though there were experimental methods for giving average molar masses for a sample, it was very difficult to know what the whole distribution was like. So what you tended to do is you would physically fractionate the sample using a solvent-non-solvent mixture, isolate a load of samples which you hoped were different in molar mass from your parent sample, use some technique to measure the average molar mass for each of those, and then try to reconstruct the original molar mass distribution, which could take you a couple of months. But with this chromatographic method these days, sometimes in just a few minutes you can get the same information. This really is a very powerful technique. And until the late 1950s, early 1960s when this was developed, it was very hard to determine a molar mass distribution. Now, well, it still has problems, but it's much, much easier. Okay, so what's involved? And the apparatus, if any of you, how many of you have actually done or used HPLC, high performance liquid chromatography? Not many, surprisingly few of you. Did you do it in a company or something like that? Sorry? In industry, yeah? It's a funny thing, if you go into industry and you go and look at an analytical laboratory, then usually what you'll find is loads of HPLC and loads of GC. Liquid chromatography, gas chromatography, as far as the eye can see. You come here, and we use it remarkably little. But you can learn a lot about spectroscopy, which most industrial companies have hardly any of. Because actually our problems tend to be different. In a university setting, we're always making new molecules. We want to characterize the molecules. In an industrial setting, you're normally trying to characterize a mixture or show that something is the same as the last batch was. And if you're in quality control, or you're working with a narrow range of things, then chromatographic methods are very, very powerful. If you're always making new molecules and you have to develop a new method for every molecule, then it's really hard work using chromatography, and spectroscopy is often more useful. So industrial labs tend to be different from academic labs in that sense, in the emphasis of different techniques. But is that right? You go into industry, lots of chromatography. And yet on our course, you hardly use it. But those of you who go out, or went out in your, where are we? Some of you will have been out in your third year, won't you? And very likely you will have used HPLC. And if so, you will all of this will look very familiar, because nowadays the equipment that is used for gel permeation or size exclusion chromatography is essentially the same as the equipment that is used for HPLC. The difference is what the column is packed with. So what do we have? Well, it's liquid chromatography. We have a liquid, a solvent, which is going to be pumped continuously all the time through our column. And if we're dealing with polymers, then that solvent is very often tetrahydrofuran, which is a THF, a very useful solvent which dissolves many polymers. Sometimes it might be something else, chloroform, toluene are ones we commonly use, and occasionally you might use an aqueous medium, but then we've got other complications that we won't mention, particularly on this course, or only briefly. That solvent, we'll degas it. We want to make sure that little bubbles of air aren't going to come out of it. And usually there'll be a little sort of filter. Sometimes it'll go through a degasser. Then we have a pump to keep that solvent pumping through the system. And we want a very even flow rate. We don't want a pump which is going to create sort of a flow profile that does this. We don't want pulses of flow. So to even out the flow rate, it'll usually be a dual reciprocating pump. That actually has two heads which work together. So when one's pushing, the other one's pulling. And by getting it working just right, you can have a very even flow rate over time. Now, we're going to have our sample. In our case, it's going to be a polymer sample. It's going to be dissolved in a sample of the solvent that we're pumping through the columns. Usually we'll want to use the same solvent, because if there's even a slight difference in how much dissolved carbon dioxide or something goes in there, that can affect what you see out the other end, because those differences will show up. But we'll have a solution. We're going to, at some point in time, we're going to inject a solution into our system. And then we're going to measure time from that point where we make that injection. And of course, the solvent is flowing the whole time through, so once we inject it, the sample that we've injected, the solution of our polymer, is going to be swept into the system where we have a column, or quite often a series of columns. Because one column will only separate over a certain range of molar masses, for reasons we'll see in a moment. And so often what you will do is you'll put several columns together to extend the range of molar masses that we can separate over, if we want good resolution. Or nowadays we might use one column with actually a mixture of beads inside to try to get a broad range. But with one column you may get a broad range, but with relatively poor resolution. So it's a compromise. If you're pumping solvent through, along comes your sample, you need to detect what's coming out the other end in somewhere. You need to know when something's emerging, other than pure solvent. And when we're dealing with polymers, now if you've used your HPLC, chances are you use something like UV detection. Would that be right? Yeah? The problem with most polymers in most solvents is either the polymer doesn't have a suitable chromophore, or more often the solvent does, so the absorption from the solvent will obscure that from the polymer. So we actually don't often, occasionally we do, but we don't often use UV detection. We're more likely to monitor the refractive index of the solution as it emerges. Because most polymers will have, in solution, a different refractive index to the solvent, and therefore you will get a response. Just occasionally you may have a problem where your polymer in that particular solvent at that particular temperature just happens to match the refractive index of the solvent. And then it's invisible. So you've got to change the solvent or do something else before you will see it. But usually, for most polymers in most solvents, a refractive index detector will enable you to monitor the concentration of polymer as it comes off the column. Sometimes we'll add other detectors. Nowadays we might have a viscometry detector, a light scattering detector, as well as a refractive index detector, and I'll say a bit more about that tomorrow. And we need to record what's going on. Now when I was a student, GPC columns were this long short, they were this long. And we had good old-fashioned chart recorders with paper that we had to mark up. Nowadays, of course, everything's attached to a computer, and there is software to at least try to analyse the data. It's actually not that easy getting software to analyse QPC data for various reasons, and a lot of the commercial software contains errors. Be warned, just because a company sells you something doesn't mean it works properly. Okay, so what's going on? Let's try to picture what's happening. So this is a schematic diagram. Don't take it too seriously. But what this is meant to represent is a column, and what we have inside this column is packed with little beads, and the beads are effectively porous. So often the beads are made of cross-linked polystyrene, which swells a little bit in the solvent. So there is solvent contained inside the beads as well as solvent around the beads. And that's what we're schematically trying to represent here. But of course not to scale. You've got to remember we've got millions of beads inside here, and just so we can see what's going on, I've drawn them as though the beads are much, much bigger than they really are. So this is meant to represent the column, this is meant to represent a bead, and what we need to understand is that solvent can flow between the beads in what we're going to call the void volume, but there is also solvent in little pores, in little spaces inside the beads in what we're going to call the internal volume. And it's the distribution between that void volume and the internal volume which is going to be important to us. And very simple, we're going to be pumping solvent in from one side, it's going to come out the other, we'll have a detector. As I say, usually it'll be refractive index that we're detecting, and before the column, we're going to have a mechanism for injecting a solution of our sample. And we're going to begin by thinking the simplest separation we might want to do, which is an oversimplification, and then we'll go a step further and ask, well, what's really going on? But let's imagine the simplest situation where we have a mixture of just two molecules, some really small ones and some really big ones. And to see what, so we can understand the limits of the technique, and then we'll focus on how we actually get a distribution. Okay, so what we've got here, ready to be injected, we've got some really big molecules which are big purple splodges, and some really little ones which are little red splodges, if you can distinguish those colours. So you've got little molecules, big molecules all mixed together, and at some point we're going to inject that mixture into the flowing stream of solvent. Okay, and we're going to be monitoring what happens at the detector over time. So we can plot it against time, or we can plot it against dilution volume, the volume of solvent that's come through the column. And of course, if our flow rate is absolutely constant, and that's why we need a really good pump, then we can always relate time to elution volume. And typically we would work with a flow rate of about 1 milliliter per minute. Okay, so what's going to happen? Well, solvent's flowing through this, that's going to sweep our sample into the column. And now we've got to ask ourselves, what's going to happen to those different molecules? They're in solution, they're buzzing around, they have Brownian motion, you know all about that, they're always diffusing in solution. But let's imagine that these big molecules are so big, they can't actually get into the little spaces inside those beads. Which means that the only volume available for them to move around in is the volume outside the beads, what we'll call this volume here, what we'll call the void volume. But the little molecules, they're small enough, they can actually get inside the bead, so they can get into these little channels inside the beads. So at any given time, some of them will be out in the void volume, but some of them will be inside the beads in the internal volume. So what does that mean as time goes on? You know, as we start to sweep this sample through the column, the big ones, they've actually got less space to move around in because they're limited to the space outside the beads. So less volume, they'll tend to be swept along pretty much at the rate that the solvent goes through the system. The little molecules, they can spend a bit of time in the void volume, but then they get into a bead, spend some time walking around in the bead, they'll come out again into the void volume, get into another bead. So they're slowed down, there's more space for them to move around in. They follow a longer path length through the same column. And so we start to get a separation. And as we proceed through the column, our big molecules, which are contained only in the void volume, will tend to move ahead of the very little ones, which are popping in and out of these beads. Until eventually the big molecules arrive at the detector, we get a response, which corresponds to the large molecules, that's swept out, and sometime later the little molecules will follow on. So that gives us an idea of how we can separate really big molecules from really little molecules. That isn't the whole story. That doesn't explain how we get a molar mass distribution. So if you're asked a question in an exam, how GPC works, don't just tell me that. That's only a bit of the story. You have to explain the whole story. So let's take a look at the bit of the story we've got so far. We've got very large molecules. They can't get into the porous beads. The elution volume, the volume at which they will emerge from the column, will actually equal the volume of liquid which is outside the beads. What we call the void volume, let's call that V with a little subscript naught. The very little molecules, on the other hand, if they're really small, they can get into the beads just as much as a solvent molecule can. And so the volume available to them, they've got all the void volume available to them, but also all of the internal volume. All of the volume of solvent which is actually contained within the pores within those beads. So there's a larger volume available. Vi plus V naught, where Vi is internal volume plus V naught gives the total volume. But that's not the whole story. That tells us why we actually have two limits, two extremes for any column. There's a point where if molecules are so big, they don't get into the column. That's one limit. And there's another point where if they're really small, they behave just like the solvent. That's another limit. And what we really want to know is what happens between those limits. How do we actually get a molar mass distribution? And this is where size exclusion comes in, where in this context, size exclusion does not mean it's like a sieve, that we have different sized holes and different sized molecules can therefore get in. If you go to Wikipedia, that's what it'll tell you. Don't believe Wikipedia. That is one way of thinking about it, but it's probably not the best way of thinking about it. So we're going to talk about size exclusion, which is actually about the behavior of molecules inside a pore in this context. Okay, so let's now try to imagine that we're inside a little space, a little pore, a little channel, inside a porous bead, inside one of these columns. And so we've got a bit of space, but that space is constrained by the walls, the pore walls around it. So this is a pore wall, this is a space filled with solvent that our little molecules can move around in. Now let's go to it. I'm going to make it simple, we're going to imagine our molecules are just spheres, at least for now. So let's say we've got a relatively large molecule. It's not so big it can't get into the pore. So we're not at that limit where the molecules are constrained to the void volume. It can get in there, but it's large relative to the size of the pore. And let's think about how much freedom of movement that big molecule has got. And remember that freedom of movement, the amount of places that we can put something, relates to entropy. So this is actually about entropy. But think about that molecule, and we'll think about the centre of mass of that molecule. How much freedom of movement is there to the centre of mass? Now, if it's coming along and bouncing around, at some point it's going to hit the wall. And of course, the centre of mass cannot get closer to the wall than the radius of that sphere, if it's a sphere. And it can bounce up there, around, hit the other wall. And so what we have is, close to the walls, we have a volume of solvent which is actually excluded to the centre of mass of that molecule. That's here what we're talking about excluded volume. Excluded means here is a bit of space that something cannot get into. If we have a much smaller molecule, it can bounce around. Again, there'll be a little bit of space near the wall that the centre of mass can't get to. But because it's a smaller molecule, it's a smaller excluded volume. If it bounces around, the centre of mass actually has a lot more space in which to move. So the effective volume that's available for movement of a molecule is larger within a pore for a smaller molecule. That is the basis, or at least that's one way of thinking about the basis of the size exclusion mechanism. There are other models in the literature. I'm only going to mention this one. So what we can say is that if we're thinking about molecules which can get into the pores, and as you'll see, we actually, in GPC, in jump emission chromatography, we can only separate, we can only get information about molecules which can penetrate those pores. Once we hit that limit of the void volume, we no longer get a separation. So the situation we want is where all of the molecules in our distribution can get inside the pores. But within those pores, more pore volume is excluded to the center of a large molecule than to the center of a small molecule. We can think about that in terms of a reduction in entropy, because we're reducing the available volume depending on the size of the molecule. So you've got a link between something to do with entropy and something to do with the actual physical size of our molecule. Now, of course, polymer molecules aren't generally solid spheres. In the simplest case, it'll be a chain-like molecule which is wiggling around and changing its conformation. We've talked a bit about that. We've said already that we can model, for some points of view, when we're thinking about hydrodynamics, how things move in a fluid flow, we can actually model that behavior in terms of an equivalent solid sphere, which is really what we're talking about here. It's the hydrodynamic volume which is important in this separation. But you can also approach it differently if you want, and if you're really good at statistical mechanics, you can go away and work this out. People have done. We're not going to do it on this course. But if we've got a chain like this, if it's unconstrained, if it's in solution, an infinite volume of solution, it can wiggle around to its heart's content. If you put a barrier nearby it, then some conformations are going to be excluded. In other words, by having a pore wall close to that molecule, you actually make some conformations unavailable. That, again, is reducing entropy, conformational entropy. So you can approach it thinking about the statistics of a chain that's wiggling around and changing its shape. But the conclusion is the same. When we put it in a constrained space between walls, we lose entropy depending on the size of the molecule. So that's the basis of our separation. That's what we mean in this context by a size exclusion mechanism. Okay. And what that means is that our separation is on the basis of the effective size of the molecule, what we call the hydrodynamic size. It relates to the hydrodynamic volume of the molecule. Okay. So if we've got a column, we have a mixture of polymer molecules. We can separate them by size inside this column. But the information we want is a distribution of molar mass. The separation isn't on the basis of molar mass. It's on the basis of hydrodynamic size. So to get a molar mass distribution, we have to have a link between size and molar mass. Between hydrodynamic size, hydrodynamic volume, and molar mass. Now how do we do that? Well, the simplest thing is just to have a set of polymers that we already know the molar mass for and use them to calibrate the column. We'll talk later and talk tomorrow about other ways of approaching it. So in the simplest case, we would calibrate our column, or our column series of columns, our system, with some samples for which we already know the molar mass. It means we need another technique, perhaps light scattering, osmometry, something else, another experiment to actually work out what those molar masses are. And usually we'll use standard samples which have narrow distributions. They'll usually be made by something like anionic polymerization. Okay. So if you've got some standard samples of the polymer you're interested in, you calibrate the GPC, we'll talk a bit about how that works, and then you can relate elution volume to molar mass. Of course, there are certain polymers for which you can go out and buy lots of standards. Polystyrene, for example. If you've made a entirely new polymer, you have a problem because you don't have the standards. We'll talk about how we tackle that a bit later. Because if you change the solvent or you change the polymer, the calibration changes. Not arbitrarily, there's actually ways in which you can understand the changes, which relate to how the chemistry, structure, configuration, chain, and its interactions with the solvents affect the hydrodynamic volume, and therefore the separation. But in general, each polymer-solvent pair will have its own calibration curve. Change the polymer, the calibration is different. So what might a calibration curve actually look for? What are we going to plot? Now, we haven't gone through the detail. I just mentioned that we had this size exclusion mechanism, and I mentioned that people who do statistical mechanics can actually look into it into a bit more detail, and people have, of course. And broadly speaking, you find that there's an approximately linear relationship between the logarithm of molar mass and dilution volume. So our calibration, our simple calibration, we plot molar mass on a logarithmic scale. So I plotted here as 10, 3, 10, 4, 10, 5, 10, 6. I could plot log molar mass, which gets into 3, 4, 5, 6. So we'd have our molar mass on a logarithmic scale, but our elution volume or time, I've expressed it as volumes here, so we can see how things relate to void volume and internal volume and total volume that I've mentioned. Elution volume or time are on the x-axis. And if you remember, we said, if we're thinking in terms of volume, you know, there are some limits. There's the void volume, and there's the total volume, which is void volume plus internal volume. That is, according to our simple picture, reflects the limits over which anything can happen. Okay, so we're only going to get a separation in that range of elution volumes. So this is getting different to HPLC. HPLC, you know, if you wait long enough, something may come out. Yeah? If GPC is working correctly, then everything will appear within a certain range of elution volumes. And ideally, then it's more or less, if you plot log molar mass against elution volume, you should get more or less a straight line. Of course, real life is nothing like that. It's never quite a straight line. It wiggles. And a little wiggle can have a big effect on your results, which means defining that calibration really carefully can become really important in getting quantitative data. But it's approximately a straight line. So as far as we're concerned here, for now, it'll be a straight line. If you go away and do some experiments, you'll soon discover straight lines aren't quite as straight as you'd like them to be. But of course, there are some limits. Now, once we get to a certain size, depending on our column packing, molecules above a certain size aren't going to separate. They're all going to appear in the elution volume. And so all molecules above a certain size will appear at the void volume, at least in theory, and therefore we don't get any separation at high molar mass. And when we get down to a certain small size, everything's going to behave much like solvent until there's a limit at the other end. So our calibration curve, remember it's an inverse relationship. Large molar mass, big molecules correspond to small elution volume, but bigger than a certain elution, bigger than a certain size, we're not going to get any separation. Smaller than a certain size, we're not going to get any separation. Any given column or set of columns will only work over a certain range of molar masses. This column actually says on it, it says 500 angstroms on there. Okay, that's telling you about the sort of the rough size range that this is polymer works. And that's quite a small, that's a small pore column. So that will only separate relatively small polymer molecules. You need to go to a bigger pore packing to separate the larger molecules. So if we put down a mixture of standards, then we'll see a whole series of peaks corresponding to different molar masses. The highest molar mass is coming out first, corresponding to a low elution volume at peak. That's the elution volume here. Low molar mass samples corresponding to a high elution volume at peak. And we can use those, if the samples are narrow distribution, we can use those peaks to define our calibration. Okay. Now in your handout, you have a page which says GPC data analysis. And somebody's going to look at it and say, ah, derivatives, I hate derivatives, I don't like the maths. Don't worry. That's there for you to understand what's going on. The maths bit of it is not going to be examined. What I want to do is just talk about the pictures. But actually, if you're bright, and I think most of you are really, you can work through the maths and see how they influence the pictures. So I'm just going to talk about the pictures. But I expect most of you can work out then from the maths why the pictures look the way they are. Okay. So we've talked about different types of molar mass distribution. We've expressed our distribution in terms of a number distribution or weight distribution. We can actually express it in terms of a distribution in terms of molar mass, or as we'll see, log molar mass. And commonly it's actually that distribution in terms of log molar mass that people actually present. And we've already seen last week, if we just compare a number distribution and a weight distribution, they look different. The same sample, the picture is different. Very often a number distribution does something like that, whereas a weight distribution will go to a maximum, yeah? What you do with your data will give different results. You can plot, with the same GPC data, you can plot different things. You need to understand what those different things are and when each of them might be useful. What we get off the computer attached to our GPC equipment is a plot of the detector response, and in most cases, as I said, that is actually a plot of the difference between, in refractive index, between what's coming off the column and some reference, which is arbitrary, usually. It's a plot of the detector response against solution volume. And so we can see how the different ways of analysing the data will give different results. I've taken as an example, and this is based on some real data that a Brazilian student got a good number of years ago, some data for a polysaccharide. This is a complex natural polymer, and we can see that the GPC curve shows two peaks. Okay, so I want you to sort of see how this complex distribution changes shape as you go through each thing you might do with the data. Okay? So in terms of elution volume, we've got a big peak followed by a smaller peak. But remember, this big peak at low elution volume corresponds to higher molar mass. This smaller peak at high elution volume corresponds to lower molar mass, because remember, in this technique, the biggest molecules come through the system first. And we have a calibration. And in the ideal case, it would be a straight line if you plot log molar mass against solution volume. We're only thinking about that region where separation occurs. If we go outside that region, it's going to complicate things. I said that everything larger than a certain size will elute at the void volume. As with so many things in life, that's only partially true. With real samples, you can find things eluting before the void volume. That's because other types of separation occur. Something called hydrodynamic chromatography can sometimes occur. So with really big molecules, you can actually separate within the void volume. But it's a different mechanism of separation. You don't need to worry about that. Just be aware that whatever we say is the norm, there's always an exception. Okay. We've got our calibration. If we apply our calibration data, then of course, what we have at low molar mass corresponds to high, and remember our calibration is in terms of logarithm of m, high log m. What we have at high molar mass, high elution volume corresponds to low log m. So by applying that, and in your handout you've got the equations, you can do it and work it through for yourself if you want, we will end up with what is a weight distribution of the log of molar mass. So we've got molar mass here on a logarithmic scale, and this is a function of the log of molar mass. And this essentially looks like our GPC curve turned around. So what was our big peak at low elution volume, that's now our big peak at high molar mass, high log molar mass. And what was our little peak at high elution volume is now our little peak at low log molar mass. Okay, so this is a weight distribution. Why is it a weight distribution? It's because we've used a refractive index which responds to the mass of material coming off. So it's a weight response. If you had a different type of detector, there was one case, when I was in industry, there was one case where we did use a UV detector, and we had a polymer which actually had a UV active end group. And so the UV response actually was giving us a number response because we had one tag per molecule. And the refractive index response was giving us a weight response. And it actually took my colleagues a little while to work out why the two detectors were apparently giving different answers, but with the right mathematical relationship between the answers. Now, so this is the weight distribution of log molar mass. That's what we normally get. If we want to, we can take that and convert it to a weight distribution of molar mass. That doesn't mean taking your Excel file and just changing this axis to a linear scale. Because if you remember this function, remember we talked about what do the shaded scripts mean when we're treating a continuous distribution. And I said it was the area of a strip which represents the weight or number fraction or component. What we've got to remember is if we stretch out this x-axis, we've got to squash down the y-axis in the right way so that the areas in a certain range of molar masses remain the same. That's why I mentioned that about the areas last Monday, because it becomes important when we think about how these handle the data. And effectively, if you work it through, in the simplest case, what that effectively means is dividing the y data through by n, as well as changing our x data to a linear scale. Again, the maths is in your handout. Work it through for yourselves if you want. So if we do that, this is what we get. So this now is the weight distribution of molar mass. And we can see that our shape has changed. When we're on a linear here, this looked like a little peak, this looked like a big peak. But as we stretch it out and squash it down proportionately, now what was the little peak looks like the really big one, because what was the big peak has got stretched out so much more because we've gone from a log scale to a linear scale. And of course, I've only got up to a certain point. This keeps going. The distribution keeps going through the wall, disappears over there somewhere. That's why we most often actually represent things on a log scale rather than a linear scale. If we can convert to a weight distribution of molar mass, we can then convert to a number distribution of molar mass. You know about that. We talked about the relationship between number and weight distribution. And if we go to a number distribution, of course, remember those little molecules, we've got an awful lot of them, although they're small. So in the number distribution, now what was the small peak in our GPC curve looks like an absolutely massive peak, because that corresponds to little molecules. We've got lots of little molecules. So we start to see how we present the data. This is all the same distribution, the same mixture, but how we present the data, our distributions will look very different. Okay, the last few minutes. Here's our problem. Hopefully now you have some idea of what goes on in a GPC column. You know that separation is on the basis of hydrodynamic volume. You know that when we use this technique, if it's working properly, then the biggest molecules will come out first, the smallest molecules will come out later. That assumes there are no adsorption effects, as I'll mention later. So it's working as we want it to work, we separate on the basis of volume. If we have standard samples which we know the molar mass of the polymer we're interested in, then we can easily calibrate our GPC system and get molar mass distributions out. And we see we can represent those molar mass distributions in different ways. As a weight distribution of log molar mass, it's a weight distribution of molar mass, it's a number distribution of molar mass, and so on. And having got the distributions, we can also calculate the averages. And again, it's in your handout. But what if you've got a totally new polymer? What if you haven't got standards available to calibrate your system? What might you do then? Well, there are a number of possibilities. Okay? Actually, what people do most often, and you've really got to watch out for this, when you read literature, you know, you're the intelligent people, always approach literature intelligently. What people often do is to use, you know, if they've got polystyrene standards, they'll calibrate with polystyrene. Even if the polymer isn't polystyrene, they will quote the average molar masses they get from a polystyrene calibration. And if they're being good, they will say these are polystyrene equivalents, or they will make it clear that it's a polystyrene calibration, because those are not the true averages for your polymer. They're what you would get for a polystyrene molecule that elutes at the same elution volume. If it's an aqueous system, then they might well have calibrated with polyoxyethylene, and they'll quote polyoxyethylene equivalent. Now, this is actually very common. Sometimes people write, even respected people write papers, and they forget to mention that this is polystyrene equivalent. Okay? So when you read it, you've got to be careful and make sure that you're the people who understand what's really going on, and you know to ask the right questions. But is that all you can do? Actually, you can be a bit cleverer than that. When I was in the industry, it's something we sometimes did, because it works quite well with, reasonably well for similar sorts of polymers, not for all polymers. But if you just take vinyl polymers, and often you're preparing different vinyl polymers, you can assume that in a similar solvent, they tend to coil to a more or less similar extent. Okay? A good solvent, as long as you're not dealing with very different solvents. And so, if you just work out what the extended chain length is, you can assume that they're more or less polymers with the same extended chain length will coil to a similar extent and therefore have a similar hydrodynamic volume. It's an approximation, but it works reasonably well at least for different vinyl polymers in remote solvents. In which case the difference arises from the different mass of a repeat unit or of a particular length of chain. So you can come up with a factor, which we used to call a Q factor, that converts from one polymer to the other, just on the basis that polymers with the same extended chain length will elute at more or less the same elution volume. That works for some things reasonably well, and it's a good approximation. For other things, it doesn't work. So what else can we do? Well, there is something called a universal calibration. If we have a parameter which actually tells us about hydrodynamic volume, we said hydrodynamic volume is what the separation is based on. If we can find something that's proportional to that, we can use that to calibrate. Okay? And it just so happens there is something that is proportional to hydrodynamic volume. If we take our molar mass and multiply by this thing, which we symbolize by a Greek letter eta in square brackets, in which we call the intrinsic viscosity, we have a hydrodynamic volume term, and we have the basis of a universal calibration, which at least more or less applies to the vast majority of polymers. We're going to talk more about that tomorrow, because tomorrow we're going to learn how you measure intrinsic viscosity, and how we can actually then use that to help us create a universal calibration for GPC, a calibration that should work for any polymer. But you need extra information. You need information about this intrinsic viscosity. Or, and I mentioned this before, we can take other techniques which give us molar masses directly. And this is something which has developed quite a lot over the last few years. For example, light scattering. In the middle of the fire alarm last week, I mentioned light scattering could give you a weight average molar mass. Nowadays, we can get light scattering detectors small enough and compact enough to put on the end of a GPC column. Actually, even when a long time ago when I was in industry, we had light scattering detectors, but they were this size on a bench, and now they're sort of this size. That you can use that effectively as long as you've got some ancillary information about concentration to measure molar mass directly, and therefore get your distribution without having to calibrate. Like a lot of things, there are complications because you lose resolution at the edges where the concentrations are low. So it sounds clever, it's not quite so easy to apply in practice, but it's something that we're doing increasingly. So finally, the final thing, just to very quickly mention, all of this method assumes that our separation is only on the basis of hydrodynamic volume. Other sorts of chromatography rely on interactions between the solute and the stationary phase. If we have any such interactions, that will mess up our separation. So you've always got to be aware of that. If you see a sample where you seem to have a long sort of tail, you've probably got some absorption effects occurring, then you can't trust your molar mass distribution. If you have charged solutes, there are particular problems. And sometimes you can have a situation where a polymer sticks to the column and then reappears a few samples later. And suddenly this peak magically appears out of nowhere. These are all problems you may encounter in practice. So you need to be aware of the limitations of the technique, the things that might go wrong, as well as understand how it works in order to interpret what you hit when it goes right. So tomorrow we'll be talking a bit more about excrometry. Don't forget, you should all have a copy of some questions to have a go before next Monday. You can now answer question 3, which is a very easy question about calibrating GPC. And tomorrow we talk about dilute solution viscometry and multi-detector GPC.